ونفس اللينك يسطا كل يوم.
Hello.
Hello.
Yep. May I start?
Okay.
So, Mr. Rashro, are you here? So I can start directly. Hello. Can you hear me?
Yes, sir.
Okay,
Yes,
I start. Okay. Let's begin. So this the sixth speaking. It's also the last speaking from our Chinese group. So let's get back to the inflammation today. In last lecture, I introduced the chronic inflammation and chronic disease. I introduced this topic to you. So today I would like to introduce how to find anti-inflammatory molecules, how to find anti-inflammatory molecules to you today. So the topic is discovery of new anti-inflammatory molecules from natural products. So how to find the new anti-inflammatory molecules? We can find different ways. We can find different ways. We have different ways from the natural products and from the rational drug design and from like the target-based drug design, different ways. But today, and it's a big direction is discovering the molecules from natural products, because natural products is a very big bank of active compounds, of bioactive compounds. So it is also a very big direction in our research group. Okay, I share the slide. So as I told you last time, the acute and the chronic inflammation mediates many disease. So inflammation is a very important pathological process in the development and the genesis of disease. So can we find new drugs targeting inflammation for the treatment of a series of inflammatory disease? This is what we consider. What we can see. So we can see the stimulators activate pro-inflammatory signaling pathways and then induce the cytokine production, and then resulting in the inflammatory injuries in different organs to cause the disease. So can we find new inflammatory molecules, compounds, and agents? So in my research group, we have three big directions. The first one is to identify new mechanisms and targets for inflammatory genesis and regulation. This topic is what I told you in last lecture. So today we focus on how to find new anti-inflammatory molecules. In this topic, in the left. So as I told, the natural products are excellent lead compounds bank, a bank of excellent lead compounds, because the natural products have a very big diversity in structure and in activity. And nowadays, more than 30% clinical agents are derived from the natural products. We can see many clinical medicines using the natural products or natural product derivatives. But the natural products themselves, they also have some shortage. Also had some shortage, like the weak PD and the PK, pharmacodynamics and the pharmacokinetics, and also the second question is unclear mechanism and unknown target of the natural products. So natural products can be very good lead compounds and can be used to find new candidates, new derivatives with new structure, new mechanism, and new targets for the development of, for the discovery and the development of chemical drugs. So we can use the chemical modification, and we can use the target discovery and validation to resolve these two scientific problems. scientific problems from the natural products. So in our group, we take the natural products as a lead compound, and we carry out the natural products based medicinal chemistry and the targeted discovery to find active molecules, especially the anti-inflammatory active molecules. Our goal is to provide new agent and new therapeutic targets for the treatment of inflammatory disease. So today I want to introduce two research stories to you. The first one is anti-inflammatory molecular discovery from the natural curcumin. So curcumin is a very famous natural product because the curcumin is from the curcuminoid sources from many herb or many natural plants like the curcuma, rhizome. So many countries, people in many countries eat this plant, this natural herb, and the curcumin is a major component in this plant. So this is the structure of curcumin. It's a very beautiful chemical structure. And before we research curcumin, so curcumin has been reported to possess many bioactive properties like anti-inflammation, anti-cancer, antioxidation, antibacterial, antivirus, and also cardiovascular protection. So also curcumin has been reported to to affect different signaling pathways and affect the different the signaling proteins. So this is a review in curcumin's pharmacology. So many signaling proteins has been affected and regulated by curcumin. So curcumin has a multifunctional natural product. Also, curcumin has been used in clinical trial. Nowadays, over 100 clinical trials of curcumin has been performed in different countries in the world. But very unfortunately, no any clinical trials succeed. No any clinical trials of curcumin succeed. All the clinical trials of curcumin failed. Why? It's because due to the very poor metabolism profile. The poor metabolism profile limits the clinical application of curcumin. For example, with an oral administration of 1.2 grams per kilogram per day for 14 days, the blood level of curcumin is very low, still below 40 nanomole, and with oral administration, a very big amount of curcumin, but the curcumin blood concentration, of curcumin is under the detection limitation, under the detection limit. So many examples. So the curcumin has a very big very big disadvantage. It's the very quick metabolism and a very fast metabolism and very low bioavailability. Why the curcumin is so chemically unstable? It's because the curcumin can be quickly metabolic because the curcumin will be quickly metabolic by in vitro, because there is a quick metabolic pathway of the curcumin. The decatone enzyme will catalyze the curcumin to degradation, to degrade it to decomposed. So the curcumin cannot accumulate a very level in the body. So we can conclude curcumin is a very bioactive compound, no significant side effects, but its clinical application was it's limited. It is significantly limited by the metabolic defects and very low stability. So how to structure and how to modify the structure of curcumin to improve its pharmacokinetics and pharmacology. This is a very interesting topic for our group. So we take curcumin as a lead compound to design and synthesize a series of analogues of curcumin to resolve this problem. And to find good anti-inflammatory molecules. So the first step is drug design for curcumin analogues. We want to get a structurally stable skeleton. This is unstable curcumin. So because the central decatone is unstable moiety, is an unstable group. So we changed the central decatone group to change to the stable, the monocarbonyl group, and to design this stable structure. So we designed a series of monocarbonyl analogues of curcumin. So in this structure skeleton, we change the different moieties, different substituents in the benzene ring, and we use different central linkers, and also we use different aromatic groups to change the different moieties of the curcumin analogues. So then we designed a series of analogues with a stable structure. After design, then we have to synthesize this compound using a series of the organic chemical method, organic chemistry. This is the synthetic design and the synthetic rules and the different synthetic rules. This is also the organic chemistry and the synthetic rules. So after then, we get over 200 curcumin analogues with this chemical stable structure. So after we get this curcumin analogues, we tested the chemical stability in vitro, and we also tested the pharmacokinetics profile in the mice. So we can see in vitro chemical stability, the degradation curve of curcumin is very quickly. The degradation of curcumin in the buffer is very quickly. But the monocarbonyl analogues of curcumin, the degradation is very low. The distribution of degradation rate of monocarbonyl analogues are lower than curcumin, indicating that this monocarbonyl curcumin analogues are much more stable than curcumin itself. Also, we tested the pharmacokinetics profiles in rats and the bioavailability, and we can see the curcumin analogues, the representative curcumin analogues has a much better pharmacokinetics profile than curcumin itself. So from these two, and this is also some PK index, So this results from the in vitro stability studies and in vivo pharmacokinetics studies indicated that the stability of this monocarbonyl analogues was greatly enhanced in vitro, and that the pharmacokinetics profiles are also significantly improved in vivo. So after we get many chemically stable analogues of curcumin, we want to further evaluate the anti-inflammatory activities. So then we do the anti-inflammatory evaluation and pharmacological study of these stable analogues of curcumin. We use the LPS, the endotoxin induced cytokine overexpression in macrophage. We use this system. We use this method, this system, to evaluate the anti-inflammatory activities. So if the compound is anti-inflammatory, this compound will reduce the inflammatory cytokine production in macrophage, in macrophage challenged by LPS. So we can see many analogues showed inhibition against the LPS-induced TNF-alpha and interleukin-6 release in the cultured mouse macrophage. So this result has been have been published in different our previous papers. We find many curcumin analogues have the anti-inflammatory activities. Also, we analyzed the SAR, the structure activity relationship. The structure activity relationship. We analyzed the SAR for these analogues. We can conclude which group, which moieties, which modification will enhance the inflammatory activities of the analogues. So this SAR and the QSAR analysis contributes to the further drug design and further modification. So more than 40 compounds for the multi-stable curcumin analogues showed promising anti-inflammatory properties. So this is the sum of curcumin analogues. This is the sum of the anti-inflammatory compounds derived from curcumin. Also, we published this compounds from in different papers. So then I want to focus one curcumin analogues to introduce how we further study this new compounds, this curcumin analog, and then how we find its pharmacological effect, mechanism, target, and then how we want to do the preclinical evaluation and to do the drug development on this compound. So C66 is an analog of curcumin. This compound has a very good anti-inflammatory activity in the macrophage and in macrophage and in vitro in mice. And also C66 has a good bioavailability and a good PK profile, The oral bioavailability is enhanced to 35%. Is much higher, much better than curcumin. And then we use the in vitro and the in vivo system to evaluate the anti-inflammatory and the pharmacological effect of C66. In vitro, we use the macrophage, renal cells, cardiomyocytes to evaluate the C66, the effect. And in vitro, we use mouse, rat, and use the HG-induced the inflammatory diabetic complication. So we can see C66 significantly inhibits high glucose, the high concentration glucose, like the hyperglycemia, in vitro. The high concentration glucose induced inflammation. C66 reduced the production of TNF-alpha and nitrate in high glucose stimulated mouse macrophage, and also it also inhibits high glucose induced mRNA expression of some adhesion molecules and chemokines in the tubular epithelial cells, So then we found that C66 can inhibit the high glucose induced the inflammatory signaling pathway, like the JNK and NF-kappa B, And also we demonstrated the JNK may mediate the diabetic inflammation and benefits effect of C66 in the renal cells. So then we focus on the JNK because the JNK inhibition is the most obvious in the anti-inflammatory actions of C66. So we focus on JNK. We take JNK. We consider JNK as a potential target of C66 for its anti-inflammatory actions. So could C66 inhibit JNK activation in other cells or in other conditions? Yeah. Then we can see that C66 can inhibit the high glucose induced JNK activation. Also, TNF-alpha induced junk activation. Then we conclude this pathway. Junk phosphorylation mediates both inflammation and cell apoptosis in cardiac cells. And the C66 also can reduce the cell apoptosis and the caspase activation in the cardiac cells exposed to high glucose. And then we conclude, we hypothesize junk may be a direct target of C66. Then we do the molecular docking and molecular docking stimulation. The docking and stimulation analysis indicates that the C66 directly bind to the pocket of the junk protein, and then we use the recombinant junk protein to test the direct interaction. We show the C66 directly inhibit JNK2, especially JNK2, while non-ATP competitive patterns, and also the inhibition against the JNK2 is selectivity to over other kinase. So almost finally, we showed a schematic illustration indicated that the protection of C66 against the high glucose induced inflammation and apoptosis is mediated by junk inactivation. Is mediated by junk interaction, inhibition, and inactivation. So the junk is a direct target of C66 for its anti-inflammatory and activity. And then we tested if C66 can protect the heart and the kidney in diabetic models, mouse models. So we can find oral administration of C66 can attenuate the diabetic complications in vivo. In type 1 diabetic rats, we can see the C66 administration did not affect the blood glucose level and the body weight, but reduced the serum creatinine and the kidney body weight ratio in diabetic rats, protect the kidney in diabetic rats, and also the pathological analysis also showed C66 significantly improved the histological abnormality and fibrosis of diabetic kidney. And also in type 1 diabetic mice, we observed the similar result. C66 protect the kidney from diabetes in mice. We also tested the heart. We also examined the heart. We find that C66 also reduced the protect the heart from the diabetic mice. This is also the heart evaluation result. We also tested the heart function. We find this the diabetes in mice induced the heart dysfunction, but the C66 administration reversed the cardiac dysfunction in mice. So finally, we can conclude this: the original drug design from Curcumin. Firstly, the structural instability to structural stability. And then the pharmacological enhancement. So the original drug design from Curcumin leads to the discovery of C66. This is a new candidate for the treatment of diabetic complication via direct and non-ATP competitive JNK2 JNK2 inhibition. So this C66 directly inhibited JNK and then protect the heart and kidney in diabetic mice. Can be so this work has been published in a very famous journal, Diabetes, in 2014. So after we published this paper, the Diabetes editor gave us editorial comments. They said this translational study presents a new target for the treatment of the diabetic cardiomyopathy. The application of a novel curcumin analog in the management of diabetic cardiomyopathy. So this C66 is a very good candidate for the diabetic complications and has been entered the preclinical study as a first-in-class new candidate. So for C66, we found the anti-inflammatory C66 in the first time in 2009. In 2009, I published the first paper on C66 in 2009, and we get the first patent in China in 2011. And then we published 4 papers on C66. And then we get this compound. In new drug development from 2015. So now we already have 5 patents on C66, protecting its molecular application and its formulations. We already finished almost all preclinical evaluations and will apply to the Chinese FDA. In this December for approval of the clinical trial of C66 treating the diabetic nephropathy. So we are looking forward to the application, to the clinical trial application, and to the result of C66, to the outcome. So that's the first story, and the second story is the anti-inflammatory compounds, molecular discovery from the chalcone, from the natural chalcone. So natural chalcone is it's a kind of natural structure from different fruit, like the orange, like the grape, orange grape, and in this plants or fruits, there are many natural chalcone structure molecules in this fruits. So the chalcone is a popular natural skeleton in plants, and this kind of structure, this kind of compound, has the high safety and also they are very active in anti-inflammation. But the problem, the question of the research on this compound is unclear, the structure-activity relationship, and unclear target. So in about 2014, we started to study this Chalcone structure as an anti-inflammatory molecular discovery, and we modified the Chalcone structure. Use different benzene ring substituent, and we synthesized a series of Chalcone structures, Chalcone compounds, more than 400 compounds from the Chalcone structure, and then we evaluate the anti-inflammatory activities in LPS challenged macrophage to see if this compound can inhibit the TNF-alpha and interleukin-6 production in the macrophage. And we can see as this panel B, we can see many compounds, many Chalcone compounds can inhibit the interleukin-6 production in LPS challenged macrophage, especially, and also we analyzed the SAR and the QSAR, and especially like these two, 23 and the 26, compound 23 and the compound 26. This compound we can see in the septic LPS-induced septic mouse model. After LPS injection in the mice, the mice will die. The mice died after 120 hours after the LPS injection, but over 70% or 80% mice survived in the LPS induced septic mice. This survived mice was treated by these two compounds. So more than 11 anti-inflammatory Chalcone-derived were discovered, were found, and then one preclinical candidate for sepsis and acute lung injury has been evaluated in our group, and maybe we are planning to submit the clinical trial application in 2023. In 2023, we are performing the preclinical evaluation of one candidate for sepsis. And this is another Chalcone structure. We modified the Chalcone skeleton for the anti-inflammatory activities. This compound 2I, is two Chalcones combined together. This compound 2I protects 100% septic mice from death, from LPS induced death, and also this compound also attenuated the LPS induced acute lung injury in mice. in mice. So we found this active compounds from the Chalcone structure. But what is the molecular target? What is the molecular target of this compound? This is a big question. This is a big question for our study. So we know this compound, we know this Chalcone derivatives. They have many very good anti-inflammatory activities, but we don't know the anti-inflammatory mechanism, and we don't know the direct molecular target. So this is what we want to do next. So for example, we take this compound. This compound L6H9 is the compound 23, is the 26. We show this the discovery of the direct target of this compound to you. This compound is a very good anti-inflammatory compound, and protects 8% mice from the LPS-induced septic death. So we list the LPS-induced inflammatory signaling pathway. We list the LPS-induced inflammatory signaling pathway here, and we know so how to discover the direct molecular target of this compound. We know this compound L6H9 can inhibit the sepsis, and then can inhibit the TNF-alpha, interleukin-6, and other cytokine expression. And then we also find that this compound can inhibit the NF-kappa-B activation, can reverse the I-kappa-B degradation and the NF-kappa-B activation. Also, we find that this compound can inhibit the MAP kinase ERK P38 junk activation in LPS challenged macrophage. So we say this compound can inhibit all the downstream signaling pathways in the LPS inflammatory pathway. So we tested if this compound can directly inhibit the signaling proteins in cell-free system, and we find interestingly, we find that this compound could not interact MAP kinase and IKK directly. So it means that molecular target of L6H9 is in the upstream signaling pathway, So then we tested if this compound can directly interact the TLR4 and MD2 proteins in the upstream signaling pathway. Interestingly, we find that this compound can interact with MD2 protein directly. Can interact with MD2 directly. We use the SPR analysis assay, and then we use a series of biochemical experiments to validate this conclusion. We found that L6H9 could not inhibit TLR2 mediated inflammation, because the TLR2 mediated inflammation it's not MD2 dependent. And then we found that this compound can inhibit the complex formation of TLR4 MD2. And it also inhibits the direct binding of recombinant human MD2 protein to LPS. And also it inhibits the interaction between LPS and recombinant human MD2 proteins in the cell surface and in the cell-free system. So this series of experiments validated the molecular target of L6H9, it's MD2. And then we used the molecular docking and stimulation reveals that the critical amino residue is R19 and Y102. Mutation of these two animal residues can totally abolish the binding of compound and the MD2. So these two amino residues are very important for the MD2 compound interaction. Then we knock out the MD2 protein. We can find knockout MD2 gene in the mice, protect the mice from LPS induced death, septic death. So we say L6H9 exacts its anti-inflammatory while targeting MD2. This compound from Chalcone, this Chalcone analogues is a new MD2 inhibitor. It's a new MD2 inhibitor. And then we further studied different other active chalcones, other active anti-inflammatory active chalcones. We find that the chalcones analogues L48H37 and L6H21 are both MD2 inhibitors, and they also possess the anti-inflammatory activities by targeting MD2, and so we can see MD2 maybe important the target of a series of anti-inflammatory chalcones. So from nowadays, we already identified 5 chalcone anti-inflammatory chalcones as MD2 small molecule inhibitors. And we also do some research using the natural products, like the first work published in British Journal of Pharmacology. We found the schematic. We found the Chalcone is a natural product. We found that this natural product also inhibit the MD2 and then prevents LPS-induced acute non-injury. And also we found the baccarin is also a active natural product. Also baccarin also target and inhibit MD2 and then protect mice from acute non-injury. The third study is focused on another natural product, also. The osler also protects against acute lung injury by inhibiting NF-kappa-B independent inflammation. We can see these three natural products have similar chemical structures, and they all of them have a very good anti-inflammatory activities. And in these papers, we demonstrate the pharmacological effects, and also we illustrated the direct target for the pharmacological effects. So summary, the natural product are very good lead compound, and also we can use the natural products to do many pharmacological research. The natural product based the medicinal chemistry, and we can find using the natural product based medicinal chemistry can find many good derivatives from the natural products, and to find good preclinical and clinical candidates, and for the drug discovery from the natural products. And also we can find the direct target of the natural products. So in our group, we studied 15 natural products, and we found the 5 direct target of this natural products. But of course, different target to different natural products, different target, and we synthesized almost 800 analogues of this from this natural products, and applied. We authorized more than 9 patents for this derivatives, for this analogues structures, and 3 of them have entered in the preclinical evaluation, and the one C66 treating diabetic nephropathy has been almost finished all preclinical evaluation, and will enter the clinical trial in the next year. So from active natural compounds, from the natural products, we can get the active compounds, and then we can do the mechanism and the target validation. So to do the structure-based drug discovery, to find the new candidates. So this is the my group. I also thanks the funding and my group in medicinal chemistry and in pharmacology and biology, and the Jianpeng in the drug development, different aspects in my group. I thank them, and thank you everyone. Yeah.
Thank you so much.
Okay, thank you.
Thank you. It is a very good and interesting lecture. I hope it will bring for our students new knowledge. Thank you for your lecture, Professor.
Yeah, thank you. We also hope to do some collaboration with university and the professors. Okay.
Okay.
Keep in touch. Okay, keep in touch. Okay. Bye.
Nice day. Bye.
Mm.
Thank you, everyone, for attending.
Thank you, everyone. Yeah.
Bye.